All right, yeah. So if you could just introduce yourself.
Sure. I'm Rodrigo Ledesma Amaro. So I'm a lecturer in the Department of Bioengineering at Imperial College London. And my research group is interested in metabolic engineering, synthetic biology, and how we can manipulate cells to make them do things that they don't do naturally. And this is what metabolic engineering is about. And we are interested in sustainability and making better products that don't rely on petroleum.
Right. So you mentioned metabolic engineering is about sort of changing the behavior of microorganisms. How exactly do you do that?
So maybe I should start explaining briefly with what metabolism is, just for those that might not know. So metabolism is basically all the chemical reactions that takes place inside a cell, and they basically convert the nutrients or the food into energy, into other chemicals or building block to build more cells. And in metabolic engineering, what we try to do is to manipulate all this chemistry and change these metabolic pathways, which are these series of reactions that convert some chemicals into others, in order to make cells produce a specific type of chemical that can have an application in the end. And that chemical can be a biofuels, a material, pharmaceutical, etc. So how we do it? So basically, because all these chemical reactions, or most of them, are carried out by enzymes. These enzymes are proteins most of the time, and these proteins are encoded in genes in the genome. So by changing genes in the genome and regulatory regions of the genome, we're going to tune in the end how much of these enzymes do we have, or if we have new enzymes, if we take genes from different organisms and we transfer them into others. So we use a lot of synthetic biology tools in order to achieve that.
Is it always genetic engineering, or are there other approaches to metabolic engineering?
So there are many approaches, and originally metabolic engineering was more about achieving this changing without genetic engineering, because basically we didn't know how to do that, and it was a lot about mutagenesis and evolution, and just leaving nature to do the job for us. And we still do and use more and more these evolutionary engineering technologies that allows us to do things we don't really understand. So it's the difference between what we call rational metabolic engineering, when we really understand all the pathway components and we decide, okay, this is the part we need to enhance or we need to remove, versus when the pathway is very complex, we don't understand well, and we leave nature to do the job. And there are a lot of in-betweens, and these things can be guided by mathematical models as well. So in the end, there is a lot of different ways to approach how we change the metabolism of cells.
Right.
So I'm just curious, so with the sort of evolutionary or the sort of directed evolution approach to metabolic engineering, could you explain this a bit more? Because I'm not sure I understand fully. Yeah,
sure. So there's this technique that has been around for a long time, but now it's getting more popular. It's called adaptive laboratory evolution, and it's about using evolution to adapt the strains to a specific condition. That is the condition can be the production of a specific things you want, or the tolerance to a specific toxic compound, etc. And how is this achieved? Basically, by evolving, or by having a specific cell in a specific environment for a long time, the cell is going to start adapting to it by normal mutations. And in the end of the process, when you do like a lot of generations after, the cell that emerged from that is very different, or at least a bit different from the one that started the process through this mutagenesis and normal processes. And and this is happening because we have a selective pressure. So let's say, for example, you want to evolve a bacteria cell to grow into a carbon source that is very cheap, for example, let's say xylose. And your bacteria cannot really grow really well. So at the beginning, it's going to grow really slowly, but the granddaughter of that bacteria, after like several generations, have like getting better at doing that. And then the strain we have at the end will be better at doing that, but we don't know what happened in the process. So then we can sequence the genome of that bacteria in the end, compared with the genome of the bacteria before, and we can learn what happened, what are the pathways that have changed and why, and we can do reverse engineering and then create strains from scratch using what we learned from that evolution.
Would you say that? Would you say that the sort of the evolutionary pathway method of you doing metabolic engineering is sort of not less effective, but would you then once you figure that out, would you then just use the synthetic mode of metabolic engineering?
So it's a good
for
question.
This pathway?
Yeah, it really depends case by case. So I would say it's very interesting to specifically, like, I'm an academic, so I work at university. We are interested in building new technologies, but also learning from them, because if we learn, we can apply this to other things as well. So I think for me it's interesting to go back to learn and to see what happened there, and if we create from scratch, the same behavior means we understood what is happening. In industry is maybe a different story, and if you have an evolved strain that works really well, you don't care what happened in between, and you just continue with that strain, adding more modification or more evolution.
And with which kind of strains or organisms are you currently working on in your projects?
So we work with microorganisms. We work with yeast primarily, I would say, but also with bacteria. So in yeast, we work with the most used yeast, Saccharomyces cerevisiae, baker yeast, mainly because there's a lot of tools and we can engineer it easily, and we can create new tools, and for that is very, very useful. But we also work with what we call non-conventional yeast, that they have specific properties that make them very good at certain things. And for example, we work a lot with a yeast called Yarovia lipolytica, which is very good at producing lipid-derived compounds, but also of other products that come from acetyl-CoA, which is a central metabolite, and basically is really good at being grown at large scale, so you can produce high amount of cells from cheap substrates. So it has properties that make them very interesting for industry. So we work with that as well. In terms of bacteria, we work with E. coli, also as the most studied bacteria, because we have all the tools, etc. But we have also other projects going with Bacillus subtilis or Pseudomonas putida as well.
How much do sort of the technologies that you make or the discoveries that you make apply across species, or are they quite independent?
So it depends on particular aspects. So we have projects where we use the learnings of one into the other, and we for example, we express the same pathway in different organisms, and we see which one works better. So I think in metabolic engineering, one of the key aspects is select the right host for your application. And that's not always obvious, and it sometimes goes through testing different ones and see which one works better. So we've done that, and many of the things are translatable, but not all, and especially if it's easier to translate or to take things from prokaryotes and put them in eukaryotes than the other way around, because of obvious reasons.
Right. Yeah. Do you think there are technologies or methods or techniques that have sort of allowed the field to progress in the past, few decades maybe? Are there any that stick out to you as quite important?
Yeah, definitely. As it's a growing field and it's changing constantly. Like every 2 or 3 years, there's like a new technology coming that make things faster and easier. So I would say genetic engineering, like in the 90s, was the first big thing that allowed us to actually remove genes or overexpress genes, and that was the key and probably the beginning of the field of biochemical engineering and metabolic engineering. And after that, all the synthetic biology revolution has been super helpful, developing tools that are more reliable, that works well across organisms, etc. And I'm particularly thinking on these new cloning systems like Golden Gate or Gibson that allow us to build more complex pathways more quickly. And of course, CRISPR-Cas9, and being able to edit genomes or inhibit or activate expression of genes with CRISPR-I, CRISPR-A. I think these technologies have really make a big difference. And the other one is systems biology, which kind of started a bit earlier than synthetic biology, and all these genomics, proteomics, transcriptomics, fluxomics. This let's say data allow us to understand much better how metabolic pathways work, how they are controlled, and if we understand that, we can also control them in our favor to do different things. And I also include there metabolic models and mathematical models of metabolism also allow us to do things that we couldn't like rationally understand, because in the end you need to think that these networks of chemical reactions, they have like thousands and thousands of enzymes taking place at the same time, and all pathways are interconnected, etc.
Right. Yeah. So is that like a machine learning problem?
So machine learning is just starting to be applied on this. It hasn't been applied a lot yet. It's more like simulations of, so far metabolic models are mainly a network, like a topology network or of the reactions that we have. They can be combined with the omic data. You can plug in proteomics data and understand things like cellular resources and the cost of protein expressions, etc. So these models are getting more and more complex, but because I think there's not enough experimental information of metabolites and fluxes, we still are not in position to use a lot of machine learning.
Right.
But it's coming and it will come very soon.
Right.
So in the eyes of like synthetic biology field, why would it be convenient to engineer non-conventional organisms?
So the main reason is because of their natural abilities. So for example, baker yeast is very good at converting sugars into ethanol because it has been selected and has been producing beer and wine for us for many centuries. But it's maybe not so good at producing other type of metabolites like lipids. And convincing cerevisiae to be able to preferentially produce lipids is a very hard work because of the many years of evolution you are trying to go against. But if you start by an organism that is already very good at producing lipids because it naturally grows in lipid-rich environment, like cheese manufacturing, for example, in the case of Yarovia, then you start with big chunk of the work already done by nature, and you just need to tweak it a little bit.
Right.
You perform experiments to try and find sort of obscure microorganisms that already might have characteristics that you're looking for.
So the question is if there is people doing that, or?
Well, I was wondering if you do, but I guess there probably will be people doing that. Do you know like how how they're performing those experiments?
Yeah, so I mean, we know a tiny fraction of the microorganisms that are there, and we know a tiny fraction of their capabilities. So we mostly go about what is published and what is understood so far, and what other people has done. And there are, at the same time, there are groups that are more dedicated to the discovery of new organisms and or characterizing their properties. On when there is a specific need, for example, like 5-7 years ago, more 7 than 5, there was an interest in making biodiesel using microbes, and then there were specific research group dedicated to identifying new organisms able to make these biodiesels, and they go through the culture collection. So these are places where like they store microorganisms for researchers that they can request them, and you get them and you analyze them and you can screen which one is better for your purposes. And at the same time, there are companies that are emerging now, and they are dedicated to go out there, to go in the wild, to go to difficult to access places, and to find new organisms with new properties.
Sounds cool.
Would you say that sort of the microorganisms can be sort of open to be manipulated in sort of like all behaviors, or is there a limit to what behaviors you can manipulate them into?
So there is a limit always, partially because our lack of understanding of the whole systems. So we often face the situations where when we start engineering a microbe and we engineer it a lot, it starts to not to be as happy as before. Like when you want an organism to express 2 enzymes, the organism is okay, it doesn't care. When you want it to express 20 enzymes, then you start to face problems. And every time you go through a round of engineering, it's getting more and more difficult to get new behaviors and new expression of genes. So that's one of the limitations that we are getting now because we have the tools to do all these like large modifications or high throughput changes. And a way around is one of the areas of my lab at the moment, which is trying to make microbial communities where you don't have one cell doing everything, but different cells, each one specialized in a specific task. And it's a little bit what happened in nature, and it's a little bit what happened in human society, right? Where you have professional people doing their own their thing really well and delivering services to others, etc. So this is where we are trying to mimic in the field of synthetic microbial communities.
And what would you apply the engineering of microbial communities to? What problems would you be trying to solve?
So one is the one we just discussed. So when you want to express a lot of different things, a very large pathway, then you can, instead of expressing the 10-20 enzymes in one single organism, you can express 10 in one cell, 10 in the other cell, in a way that they exchange the metabolite in between. So
and what pathways would you be sort of trying to express?
It can really be in any pathway as long as they can exchange this metabolite in between. But you can do things like production of specific compounds and make them more efficient in this division of labor. But you can also make them use a complex substrate, and each cell can be specialized in using different aspects of the substrate, or they can cooperate in a way that the byproducts that are undesired in the process can be recycled back and converted into the product that we want. So there are a lot of like interesting societal behaviors that we can try to engineer and to bring to these communities.
And also wondering, are there any risks with using metabolic engineering?
So, I mean, what do you mean by risk?
for instance, for in outside the in the application aspect, yeah, in the overall application.
Yeah, I guess yeah, there are risks that need to be like considered like in every scientific field, I would say, and depending on the applications, the risk will be more important or less important, or there will be no risk at all. For example, I've worked a lot in the production of compounds for industry, like making like vitamins or colorants or fragrances, and these things are very low risk because in the end you just you have always the strain contained in a bioreactor, and there's a lot of protocols in place to make sure that you dispose well the modified cells, and everything is like tightly regulated, and then your final product is separated from the cells, so there is no real possibility of like see these strains going outside. In other applications, more in the biomedical space, for example, there are risks that need to be considered. Like, I don't know, there are people engineering bacterial cells to kill cancer cells, for example. So there is a risk on introducing bacteria into the human body, and that can create immune reactions. All these things need to be studied properly, and they are studied.
Yeah, thanks. Do you have a particular application that something that you kind of stands out to you as quite exciting?
In general, in the field?
Yes.
Yeah, so I guess, I mean, there are a lot of things going on that are very exciting in my point of view. And
you can go, you don't have to choose one. You can
I
choose
can mention
as many as
a
you
few.
like.
So one thing that is, I mean, I mentioned this bacteria killing cancer, and I think the application in biomedical research is super, super cool. And all these, like I mentioned microbial communities as well. And in biomedicine, we are realizing the importance of microbiome and all the microbes we have in the gut or in our mouth and our skin are like super related to health and disease. And this, like, we are trying to develop methods and technologies to engineer these microbiomes as well, to be able to use better the food that we eat, or even to kill pathogens when they arrive, or to sense that there is an inflammation and there could be an underlying like a situation that need to be checked. In more industrial applications, I would say one that is super exciting at the moment is food. We are living this food revolution at the moment, where there are substitutes for meat, for milk, and many of them go through metabolic engineering. For example, Impossible burgers are made with some protein expressed in a yeast that then are used to get the meaty taste to this meatless burger. Or all these companies that are doing synthetic milk, and they basically recreate the components of the cow milk, but without cows in the process, by expressing these enzymes or the proteins that are normally found in milk, but expressing them in yeast. And the food area is very interesting, and there's a lot of things going on in both the creation of ingredients in a more sustainable way, but also in creation of the food substitutes. Another area that is very interesting is materials. So creating sustainable materials, bioplastics, to substitute like non-degradable plastics that come from petroleum, for example. But apart from substituting all the materials that we already have and make it more sustainable, we can start thinking on creating new properties that didn't exist in our current materials. Like, for example, these living materials that contain cells inside, and they can sense the environment and can react to it. And you can start thinking of concrete that heals itself, and when there is like a fissure in the concrete, there is some bacteria that can create some material that can fix the concrete, or even like devices that contain cells that can produce the insulin for diabetes treatment, right? And can sense the glucose in blood, and at the same time can produce what is required. So there's a lot of exciting things going on.
Yeah. And do you know a specific example, like how they, so say in biomedicine or in biomaterials or something, the actual metabolic pathways that they have engineered? Could you give an example?
So for example, let me think.
Yeah, it might be on the spot. It's okay if you can't remember.
No, yeah, I mean, I was thinking which one to choose, but
No, fair enough.
for example, I mean, we are working with, we have a project where we are building a living biomaterial. So we are trying to make a material for wound healing. So this is a cellulose material that contains some cells, and these cells can secrete some factors that help wound healing. So we have several organisms in there, and one pathway is the creation of cellulose. So the bacteria can create the cellulose material, and by engineering the pathway and the enzymes that convert the substrate like glucose into cellulose, can make the bacteria to produce thicker cellulose, or cellulose that retain more water and can keep hydrated for longer time when you put it in the wound. And on the other hand, you can also engineer that bacteria or another to produce a specific wound healing factor. And you just express the genes that, and it can be a human factor. You can take the gene from human, put into the bacteria, and the bacteria will produce that in the material.
So will that, if the bacteria produce the cellulose, will be like the material, will it be like in a way self-assembling as well?
Yeah, that's right.
That's very cool.
I think a little bit more sort of forward-looking, like future-looking, which I guess is always speculation. But how do you think that the field can progress and become sort of more in control of organisms? Like what sort of, I guess, what is holding back the field at the moment?
So I think one of the, I mean, of course, our limited tools and knowledge, and we work as much as we can and in like developing new tools and expanding our understanding of metabolism. But I think one of the main factors is legislation, and I think many companies are not investing so much in these GMO technologies because of the GMO label and the difficulty of commercializing products that come from GMOs, either plants or microbes, and that's something that we see changing in other countries. In Europe, it's still very restrictive, and something that I think will change in next years. But that's one of the reasons there is less investment in the application side, because companies very often say, "Oh, but this is GMO. I don't buy it." There's a little bit of public opinion as well, and it's totally related to that as well. So there's still like a lot of things we need to do to convince people or to tell people that this is a safe technology, that this is a technology with a huge potential to make the planet more sustainable. I guess other aspects at the end of the day is making processes that are economic. So we can do a lot of things in the lab. We can demonstrate that we can make all these biofuels, materials, pharmaceuticals, etc. But to build them or to make them in an economically feasible, affordable way, that's still a challenge. And there's a lot of research that needs to be done also in like how we do this process more efficient.
Right. Yeah. That's good.
I have a question. Well, just yeah. So let's say that legislation wasn't really as much of a problem. Like, let's say, like in the future, how could metabolic engineering, for instance, like help build the bio-based economy?
So I think now we can produce all these things that we usually make from petroleum, all these materials, pharmaceuticals, foods, additives, etc. We know how to do all this with microorganisms or with plants, and I think with a little bit of trust from the industry and like investment, I think all of that is achievable. And we've seen that with the COVID vaccine, right? As in very short time, we were able to develop a new technology that is saving a lot of lives, right? And that was allowed basically because of the will of our politicians and the money behind for pushing a technology. And I think doing something similar in other areas of application, I think we have the capacity to build all these sustainable processes.